So today's lecture is going to be on protein synthesis. Protein synthesis is how these genes are expressed. In DNA, we talked about that a gene was a section of DNA that codes for one particular protein. So now we need to look into actually how these proteins are made. Okay, so our gene expression in general, is just the process of using genes to make the cell products. Okay, that's all our gene expression is. So we're using these genes. Remember, a gene was that section of DNA that codes for one particular protein. So we're using the genes to make the cell products, which as a general rule, those cell products will be proteins. Okay, and so gene expression can be regulated by a couple of things. So gene expression, the cell itself can turn genes on and off. So the cell could say, this particular portion of DNA, it is not going to be turned on. And this particular portion, it will be turned on. All right, so the cell can turn genes on and off, because remember, the gene is the section of the DNA that codes for one particular protein. So you have plenty of genes in your body that do not code for any proteins because they are turned off. Okay, and then eukaryotic cells, remember our cells of the membrane-bound organelle, for example, us, eukaryotic cells, they can alter the messenger RNA once that has been produced. If you look at our overview picture over here, you'll see that we've got our DNA. Our process of protein synthesis goes from the DNA to the messenger RNA, which is transcription. And so a form of regulation in eukaryotic cells can happen right there. It can alter that messenger RNA. Then that messenger RNA, you'll see here it's coming out of the nucleus. Okay, that messenger RNA comes out here to the ribosome to do translation, which is the second part, or the formation of the actual protein. So the first step in protein synthesis is transcription. And so transcription is the process here of taking the DNA and turning that into messenger RNA. And in a little bit we'll look at we'll compare and contrast DNA and RNA. So in this process, though, we're taking the DNA and we're turning it into messenger RNA. Again, this is our first step in the process of protein synthesis. So when we take the DNA and we turn it into messenger RNA, the first thing that we need to do is we need to open up that DNA. If y'all remember, DNA is a double-stranded molecule, and the importance here is right in the middle. That's where the order of those nitrogen bases determines which proteins will be made. And so to be able to get that information, we have to open up. You'll see here we've opened up this DNA strand. And that allows the enzymes to get in here and get access to this DNA so that they can make a copy of it. And the enzyme that's going to do that is helicase. Remember, ASE is an enzyme? Helicase is the enzyme that's going to come in here and it's going to unwind or open up the DNA strand. Okay, so helicase will unwind, open up the DNA strand. Remember, these nitrogen bases are held together with hydrogen bonds, and hydrogen bonds are not very strong bonds. So this helicase enzyme will come in there, unwind, break up those hydrogen bonds between those nitrogen bases. And for eukaryotic cells, this will occur in the nucleus. Okay, for prokaryotic cells, this still occurs, but it occurs it doesn't have a nucleus for it to occur in. So it occurs right there along the DNA. Okay, but in a eukaryotic cell, this will occur in the nucleus. Remember, DNA cannot leave the nucleus; it's too big, and so this transcription part, this DNA to messenger RNA, in eukaryotic cells will happen in the nucleus. The enzyme helicase will unwind or open up that DNA, break those hydrogen bonds. So once that DNA is opened up, another enzyme will come in called RNA polymerase. And remember, here's our ACE, and it's making a polymer, right? So this RNA polymerase will now come in, and it's going to make, so this is our RNA polymerase enzyme right there, it's going to make this RNA strand here, which is technically a messenger RNA strand. So the helicase enzyme opened up the DNA, so that the RNA polymerase enzyme could get access to those nitrogen bases, and it could make a copy of them. Okay, and so what this RNA polymerase enzyme is doing is it's making a copy of the DNA. And you'll notice it's doing that complementary base pairing that we did before, A to T, C to G. Okay? And I know there's some U's here that make RNA a little bit different, and we will talk about those differences in a few minutes. Okay? But it's going to do that complementary base pairing to make this messenger RNA copy. So once that messenger RNA copy is made, that messenger RNA then needs to be processed. Okay? And so that messenger RNA is going to be processed by what's called a spliceosome. And that's what we have happening in this picture over here on the left. Okay? So these large orange molecules, these are our spliceosomes. And if y'all remember introns and exons from DNA, introns are the genes that are not expressed. That's the junk DNA. The exons are the ones that are expressed, EX for expressed. So we want to keep those exons, but those introns, they can go away. And so spliceosomes will cut out that intron and then join these two exons back together. So you can see here comes in so this is our messenger RNA, the top one here. This is our messenger RNA that was made based off of the DNA. It's got some introns on it. It's got this extra DNA that we don't need. Okay, so the spliceosomes, the large orange molecules, they'll come in, they'll attach. You'll notice here they're clipping that intron off, and you'll end up with those fused exons. And the intron then will be all gone. And so then all we have are exons left. And so that's what will be transcribed, are those particular exons. Something else that can happen here during this, or that needs to happen here, in addition to the removal of the introns, is we will add what's called a 5' cap, as well as what's called a poly-A tail to the messenger RNA. Now the 5' cap, that is going to help prevent degradation. It's going to help prevent the messenger RNA from being broken down when it goes out into the cytoplasm. And that will also help the ribosome and the messenger RNA recognize one another. Okay, so that's going to be our 5' cap there. It's going to help with degradation. It's going to help prevent the messenger RNA from being broken down in the cytoplasm. And it is going to help the messenger RNA and the ribosome recognize one another. On the other end of the messenger RNA, we're going to add a poly-A tail. And that poly-A tail is also going to help prevent degradation. It's going to help prevent the messenger RNA from being broken down. And it will also then play a role in how many times this messenger RNA can be translated. So how many times can this copy of a messenger RNA be used to make a protein? Okay, and so if we look here, we've got a picture of a fully processed messenger RNA here. Oh, I'm grabbing the wrong stuff somehow. Okay, let me grab the right thing. I can do it. Here we go. Okay, so what we have here is a processed messenger RNA. So you can see this is our original messenger RNA up here on the top. Okay, so this strand here on the top. You'll notice it has exons, it has introns, it's got a mix of both. Okay, and so in the messenger RNA processing process here, you'll see these introns, they've been cut out. Okay, so those introns are getting removed. So all that we have left in the final one, okay, is just exons that are going to be coded for. We have the 5' cap on one end. That again is going to help prevent the messenger RNA from being broken down. And it's going to help the ribosome and the messenger RNA find each other. And then we have the poly-A tail on the other end, which is also going to help prevent the messenger RNA from being broken down, and it's going to help determine how many times that messenger RNA can be translated into a protein. One of the things that we also mentioned on the very first slide was that during this stage, when the messenger RNA is being processed, okay, that's also one of the ways we can determine gene expression, which proteins are going to be produced. And so if you'll look here at this picture, you'll notice you've got a different combination depending upon the way this messenger RNA is spliced. You can see on the left side here, the green one has been spliced out. On the right side here, the yellow one has been spliced out, and those end up with two different proteins. So that's another way that gene expression can be regulated, is in the processing here of this messenger RNA. So let's take a second then and let's talk about what actual, the differences, and let's compare and contrast RNA and DNA. Okay, so let's talk about DNA first because we're familiar with DNA. So we know that DNA has the sugar deoxyribose in it. We know there's only one kind of DNA, the kind that lives in your nucleus, that's it. Okay, it is double-stranded and it contains a nitrogen base called thymine. Okay, RNA contains a nitrogen base called uracil. It is single-stranded. There are 3 kinds of RNA. We've got messenger RNA, transfer RNA, and ribosomal RNA. So we have 3 kinds of RNA: messenger RNA, transfer RNA, and ribosomal RNA, whereas you only have 1 kind of DNA. RNA, it can be also found in the cytoplasm, and its sugar is ribose, okay, whereas DNA's is deoxyribose. They do have some things in common, though, okay, and some of the things they have in common are some of our nitrogen bases, adenine, cytosine, guanine. You find those in both of them. So both of these nucleic acids have 3 nitrogen bases, okay, that are the same. They have adenine, cytosine, and guanine, and then RNA will have uracil, where DNA will have thymine. Okay. Something else that we need to keep in mind that is the same about the 2 of them is that both of them will be found in the nucleus. You know, DNA is found in the nucleus. Okay. RNA is found in the nucleus as well. Okay. In particular, messenger RNA is found in the nucleus. Okay. Ribosomal RNA and transfer RNA, those are both found out in the cytoplasm. So when we said that RNA is found in the cytoplasm, that's going to be the transfer RNA as well as the ribosomal RNA, and then the messenger RNA will travel out there. Okay, in the nucleus, though, the messenger RNA starts in the nucleus, and we know DNA is always in the nucleus because it can never leave. Okay, we know that both of these are also going to help make proteins. Okay, DNA has the directions, RNA is the one that actually does the work, and then they're both made of nucleotides. They're both nucleic acids, so they both have that nucleotide monomer, a sugar, a phosphate, and a nitrogen base. Now, those sugars are different, and sometimes the nitrogen base is different, but that basic nucleotide structure is the same between the 2. Okay, so let's take a closer look at some of those things that we said RNA had different. Okay, so we said RNA had 3 different types. So up here we've got some messenger RNA. Okay, single-stranded, contains uracil instead of thymine, but still made of nucleotides, still has a sugar, ribose, a phosphate, and nitrogen bases in each nucleotide. Over here, we have tRNA. tRNA carries amino acids on it. You'll notice, though, it also has nitrogen bases. And then here would be our rRNA. The ribosome is made up of rRNA. So we've got 3 types of RNA. And then here's our different nitrogen bases. Okay, so DNA has thymine. And uracil is found in RNA. Doesn't matter what kind of RNA. All kinds of RNA have uracil in them. And they just have a few slight differences. You can see right there would be our number 1 difference there between the 2. Okay? And then we lose that hydroxyl group as well. Okay. So those are some of the differences between RNA. Okay, so let's look at RNA base pairing because RNA does not have thymine like DNA does. Okay, so RNA goes A to U, C to G. Uracil just takes the place of thymine. Uracil never, ever, ever, ever bonds with thymine. Uracil takes the place of thymine. So in a strand, you would never see both of them. Like, for example, up here, I've got DNA. So I would never see a U anywhere in this strand here because I'm in DNA. The opposite side of my DNA would be a T, an A, a C, and a G because I use my complementary base pairing, and DNA does not have U's, RNA does. Over here on the right, I've got my DNA strand now, and now my DNA strand has been opened up, so I'm using my RNA polymerase to make my messenger RNA strand. So my A now goes with a U, where in DNA, where it would go with a T, the U just takes the place of the T. So the A goes with a U, T goes with an A, G with a C, C with a G. All the rest of that stays the same. Okay, T would still go with an A because A is in RNA. Okay, if you look here at this picture, here's our DNA. And so I've got my DNA with my T's instead of U's, A to T, C to G, okay, all the way through here. Okay, then when I'm making my messenger RNA here, okay, you'll notice now I have my U's, okay, and I've still got my A to T still applies. When I see a T in DNA, that I still have an A in RNA. That's nothing different. T and U do not go together. U takes the place of T. So when I have an A here in my DNA, I've got a U in my RNA. So if you look down at the bottom here, I've got my messenger RNA strand down here. So what DNA would this have come from? Well, I have a U in RNA. Well, U goes with A, right? I have a G, so I've got a C. I've got an A. Well, I'm making DNA, so I need a T, not a U. Now I have a G, so I've got a C. U goes with A. C goes with G. Okay, so now let's look at the second step of protein synthesis. So we talked a little bit about how to express the genes. We looked at our first step, which is transcription. So all I've done is taken this DNA and turned it into messenger RNA. I still haven't turned it into a protein, and that's what the translation part is. Translation is taking this messenger RNA and turning it into the actual protein or polypeptide. Now for that to happen, the messenger RNA needs to leave the nucleus. It goes out of the nucleus, out into the cytoplasm, and it's going to go find a ribosome. So translation occurs out in the cytoplasm at the ribosomes. Remember, a prokaryotic cell doesn't have a nucleus, but it still has ribosomes. They are technically not a membrane-bound organelle. So that messenger RNA that was made off of the circular chromosome of the prokaryotic DNA, it'll just move over in the cytoplasm to a ribosome. So this messenger RNA leaves the nucleus and goes out to the ribosome. And out at the ribosome is where we're going to actually have the formation of the protein. And if y'all remember, a protein is made up of amino acids. So it's got a whole bunch of amino acids. They're the monomer. And those amino acids are held together by peptide bonds. That's why a protein is called a polypeptide. So the protein, okay, those amino acids are all held together by peptide bonds. So when things join together, remember we talked about enzymes, we gave an example of anabolic reactions that build something up. An example was a dehydration synthesis. Well, the formation of the protein is a dehydration synthesis. When those amino acids join together and make that peptide bond, okay, so here's my peptide bond right here. Y'all know I can't write very well with this thing. So there's my peptide bond, okay, right there holding the two together. When those two join together to form that peptide bond, water leaves. So I synthesize something by joining these two together. Okay, there's my synthesis. My dehydration is my water leaving, okay, and that creates my peptide bond. And so a protein is made of a whole bunch of peptide bonds, a whole bunch of amino acids held together by peptide bonds. So that's where the polypeptide part would come from. Okay, so when we're determining which amino acid, because you'll notice I've got this messenger RNA strand, but there are no amino acids there. It's just a bunch of letters, just a bunch of A's and C's and G's and U's. So I need to break these into codons. And so in the DNA, I group my nitrogen bases into groups of 3. So in my DNA, you see I group these into 3s here. Okay, so in my DNA, that's a triplet. Okay, so a triplet is found in DNA. My groups of 3 in my RNA, my messenger RNA, those are called codons. Okay, so my groups of 3 nitrogen bases in messenger RNA are called a codon. Okay. Each codon, so let's look at my messenger RNA here. So up here up top, I've got my messenger RNA. So if I group these into 3s, that's not helpful. They didn't give me a complete strand here. So when I group these into 3s, those are codons. Okay. So each group of 3 here on my messenger RNA is a codon. Each codon codes for one amino acid. So each codon codes for one amino acid. So how many nitrogen bases does it take to code for one amino acid? Well, it takes 3 nitrogen bases, because 3 nitrogen bases equal 1 codon, and 1 codon equals 1 amino acid. You can see here, my amino acid here is leucine, and that's based on this codon right here, these 3 amino acids. So this process, again, is translation. And so what I have happening here is I have a transfer RNA coming over. So these blue molecules here, these are my transfer RNAs. These guys right here. Okay? And the transfer RNAs have on them, they have on what's called an anticodon. Okay, so that's what this part is here. You notice there's 3 nitrogen. What does anti mean? Against. Right? And I've got 3, so I've got 3 nitrogen bases here. Okay, on my anticodon, and you'll notice my anticodon base pairs with my codon, right? My base pairing here would be a CGC, which is what's on my anticodon there on that transfer RNA. So the transfer RNA has an anticodon on one end, and then it has the amino acid on the other end. And transfer RNAs are amino acid specific. Like this particular transfer RNA only carries alanine. This particular transfer RNA over here only carries leucine. So if it's the anticodon matches the codon, then that's how it knows that that amino acid is what comes next. Okay. So one of the ways that we can determine that, okay, is if we use what's called a codon chart. So my codon chart, you'll notice, is based on messenger RNA. So I have my messenger RNA that came out here it's headed out of the nucleus, headed out to the ribosome. So if I break it into 3, I break it into my codons. Okay, this one has 4 codons in it, so it would code for 4 amino acids. I use this messenger RNA to read my codon chart. So my first base here is an A. So I use this first side here. So I know I'm in the A row. My second base here is a U. So I use my second base, so I use this along the top. But I know I was in the A row, and now I'm in the U column, so now I know I'm stuck in this box. I can't leave this box because my first base put me in the A row, my second base put me in the U column. Well, in this particular box, I have some choices. You'll notice some of the other boxes you don't have choices. In this box, I have some choices. So now I need to look at my third base, which is a G. Well, you notice I have 4 G's over here. Well, I'm going to use this G right here because I'm in the A row. So when I follow that G all the way back, that gives me methionine. So I would do the same with my second one. Okay, so my second one here, my first base is a C, so I'm in the C row. Second one is a U, so I'm still in this U column, so I'm in this box here. And now it doesn't even matter what my third base is, right? I've got leucine happening. Okay, and I would do that all the way down. Okay, let's do them all, just so we can see how we would read this chart. Okay, so our next one is A. So I'm back in this A row. I've got G, so now I'm over here in the G column. So now I'm over here, and now I have choices. So I need to look at my third base. So I use that U, so I'm at serine. Okay, and so our last one here would be our first base is a U. Okay, so I'm in this U row. I've got a G. Okay, I'm up here. And I'm going to use this particular G because I have choices and I have to stay in that U row. So now I'm at tryptophan. So now I'm deciding which amino acids are going to go. So my messenger RNA, my transfer RNA would bring these amino acids over. Okay, so this one here is my messenger RNA. I use that to read my chart. And my transfer RNA would bring these amino acids that I determined from my chart. Okay, so the transfer RNA brings those over, and it will bond with the messenger RNA to drop those off at the ribosome. Okay, so this is another chart that we could show you. Who knows what they will give you, so I just want to expose you to a couple different things. If you look closely at this, you will notice this is not a codon chart. Okay, in this one, I've got T's. So what is this telling me? This is telling me DNA. So in this instance, if I was shown this chart on a test, I only have RNA. So I would need to take this backwards to my DNA, to a G, an A, and a T, because it's DNA, to be able to determine what this one would be. So now I've got my G, my A, and that puts me here. Okay, so let's kind of sum this process up a little bit here. So this is my translation portion. Remember, it's not all of protein synthesis. We're going to look at it all in just a second. So this is just my translation portion. So here's my messenger RNA, that red strand there. It's come out to the ribosome. That's what these large, I've got a large subunit and the small subunit. Those are the ribosomes. So now I've assembled the ribosome around this messenger RNA. And you'll notice as the messenger RNA goes through the ribosome, okay, it's grouped into those groups of 3, those codons, and the transfer RNA is bringing amino acids with it. We would use our chart to read our chart to determine which amino acid would come over. Okay? And as those amino acids come into the ribosome, they're going to go through a dehydration synthesis to make a peptide bond. So when they are hooked together with, when they are hooked together with that peptide bond, they're going to lose a molecule of water. So now this messenger RNA, if you look here, they just keep moving this way. This messenger RNA just keeps moving through the ribosome. So here's my new codon. Here's my transfer RNA that comes over with an amino acid attached. Okay, that amino acid that came in, it would attach to my growing polypeptide chain. My messenger RNA would just keep on moving over until eventually it's going to reach what's called a stop codon. Okay, and when you get a stop codon, okay, it means stop. There's nothing else that's happening after that. Okay, if we take a look at our codon chart again real quick, you'll notice that some of the codons on the amino acid, some of the codons code for stop. They don't code for an actual amino acid. So if I'm looking at my codon chart here, I've got a couple places where I'm going to code for stop. So if I see UAA, so UAA, okay, if I see UGA, UGA, I'm going to stop assembling that protein. No amino acid is added there. Nothing is added after that. Stop means stop. So if there's, like, let's say there's some kind of mutation, and that mutation codes for a stop codon. The protein doesn't keep building after that. It's done. It's over. It is stopped. Okay, so let's just kind of overview this again real quick. So we've got protein synthesis. We've got 2 steps. All right, we've got transcription is the first step, okay, which is the DNA to the messenger RNA, and translation is the second step, which is taking this messenger RNA here, okay, and taking it out to the ribosome and turning it into the polypeptide or the protein. All right, so you guys will be modeling this in class, okay, and so I will see you then.